Welcome back to The Explainer. Today, we're diving into one of the absolute most suspenseful moments inside the laboratory. Following insemination, or direct sperm injection, a really central mystery remains. Did normal fertilization actually occur? This explainer is going to take you through the very first real lab proof point, where we shift our focus away from individual gametes and start looking at early embryos.
Exactly. So, here is our roadmap for today. We will quickly cover why biology requires visual confirmation, explore the critical timing window, define the classic 2-pronuclei zygote, interpret abnormal patterns, and finally, discuss how this vital check narrows down our starting cohort. Section 1. Biology, not an assumption. Okay, let's dive into this because we need to shatter a really common myth right out of the gate. Merely placing gametes together absolutely does not guarantee success.
When we ask, did fertilization actually happen, we're recognizing a fundamental truth. Fertilization is a highly complex biological process. It is not just some procedural assumption. Simply bringing a sperm and an egg into close contact doesn't automatically mean they will combine correctly. We need distinct visual confirmation to know for sure if the biological sequence actually worked.
Right, it's super easy to just assume that sperm meeting egg equals immediate success, but the reality is so much more demanding. The mature oocyte must respond perfectly to sperm entry. A complex series of internal biological steps has to unfold before any embryologist can even think about declaring a victory. We literally have to visually confirm specific markers to prove that genomic integration has truly begun.
Section 2. The critical timing window. Finding those markers is literally a race against the laboratory clock. We only have a temporary biological state to observe here.
That's right, because timing protocols in the lab are incredibly strict. At 0 hours, insemination or injection occurs. Now, if we check too early, say around 10 hours, those visual markers might not be fully formed quite yet. Conversely, if we wait until 20 hours or beyond, the cell undergoes syngamy and cleavage, meaning the markers vanish completely. Embryologists must perform this check during a very specific 16 to 18-hour snapshot. Checking late misses the crucial evidence entirely.
There's a brilliant saying in the lab for this. The pronuclear stage is informative precisely because it is brief. It beautifully underscores how this assessment is just a quick snapshot of an actively moving, highly dynamic event. It is a fleeting window into transition, rather than some fixed, permanent state. Section 3. The classic 2PN zygote. Let's look at the ultimate success story. What specific visual elements are embryologists actively seeking under the microscope during this fleeting window?
The absolute key structure they look for is the pronucleus. Basically, this is the visible nuclear structure containing the genetic material from each parental genome right before they fuse. Once these distinct structures appear, the single cell is formally called a zygote, finally moving past the gamete stage.
And the standard benchmark for success here is incredibly specific. A normal zygote features exactly one maternal pronucleus and one paternal pronucleus. Alongside those, we expect to see two polar bodies. The expulsion of these polar bodies acts as a vital marker of proper meiotic progression. It confirms the successful transition from the M1 stage directly into the M2 phase and beyond.
Now, what's really interesting about this is how simple the core finding actually needs to be. Seeing 2PN, meaning 2 pronuclei, gives us our initial reassuring proof point. It is the absolute gold standard benchmark used universally to calculate the normal fertilization rate for any given cycle.
Section 4. Interpreting abnormal pronuclear patterns. Because, well, biology doesn't always follow the textbook. We have to address what happens when this visual check fails to show those classic 2 structures. We generally encounter 3 primary abnormal patterns, and these can obviously be tough news for patients. A 0PN finding means 0 pronuclei are visible. This indicates failed fertilization or perhaps a missed observation window. Next is 1PN, showing only a single pronucleus. This one is ambiguous and highly atypical. Finally, 3PN reveals 3 pronuclei. This points to extraparental genetics, very often due to polyspermy, which is where more than a single sperm successfully enter the cell.
And it's crucial to remember that abnormal results, like a 3PN finding, prove human biology can still falter despite advanced assisted techniques. Abnormal fertilization frequently arises even after direct sperm injection. The route of entry simply does not guarantee a perfect biological response.
Section 5. Narrowing the IVF cohort. Now we must transition to the clinical and emotional reality of cycle attrition when early development falters.
To put this into perspective, think about the total number of mature eggs retrieved. After the pronuclear check, that starting cohort shrinks significantly. We are left with only the normally fertilized zygotes, leaving behind any unfertilized or abnormal cells. It represents a very stark visual drop in available numbers.
So, the crucial point is, this specific moment sets immediate numerical expectations for the remaining journey. It completely transforms our understanding of the cycle's potential trajectory. As the quote says, the fertilization check is the first major narrowing of the IVF cohort.
While seeing a 2PN zygote is definitely encouraging, we must maintain strict scientific objectivity regarding its limitations. This early check absolutely cannot guarantee blastocyst development later in the week. It does not ensure implantation success, chromosomal normality, or ultimately a live birth. Much development remains entirely unseen.
Which leaves us with an incredibly grounding thought. A 2PN zygote is a promising start, not a finished success story. This beautifully balances necessary clinical realism with profound human hope. We celebrate the milestone, but acknowledge the long road ahead.
So what does this all mean? Well, it leaves us with one massive question. Once normal fertilization is definitively confirmed, can those newly formed, highly fragile zygotes actually survive and continue developing inside the laboratory culture system? Thank you for joining us for today's Explainer. Next time, we will explore embryo environmental engineering to see exactly what happens inside the incubator.